I'm Kyle. I know most of you. I'm an assistant professor in gastroenterology at CHOP, and I also direct the bioinformatics group for the PennCHOP microbiome program. I work with, Jaylon's not here yet, with Chunyu and Yue, who are sitting back there. Oh, and if I could, just very quickly, I have a survey for everyone. It doesn't matter so much if you love me or hate me, but it does matter if you fill out the piece of paper. And I can't collect it, so please give it to Chunyu. Would you guys pass it around, please? All right, good. So now we've got the administrative stuff out of the way. I'm Kyle. Good to see everyone. All right, here's what I expect to cover today. So I want to keep the talk down to about 35 minutes. And then I set up an interactive demo. And so if people have laptops, we can do a couple huddles and explore a little bit inside the web page I made. If not, I can do a little bit up here to kind of show you how things play out. So I want to cover today taxonomy versus phylogeny. What's the relationship? What are the differences? Classical approaches to bacterial taxonomy, from culturing and how bacteria are named. And then in the last part, go over numerical approaches to taxonomy. So actually a pretty old practice starting in the 60s, but in today's work, it forms kind of a parallel approach to naming bacteria according to well-established names, and in parallel, we numerically cluster bacteria into our own taxa that don't have names. So how are those two things related? Okay, good. I'm just going to get some terms out of the way at the very beginning. So the taxonomy is the system of boxes into which you can put individual organisms, and the taxa are the boxes. You can arrange the taxa into ranks. You don't have to, but you can. So, and the ranks are genus and family and species and class and order. So those are the ranks, the taxa, and then the taxonomy is the system. So bacteria are named under the bacteriological code. And when they established the code, they said, oh, this is totally not official. You know, it's an agreement among all scientists, but it's kind of official. It's as official as it gets. And then separately, phylogeny is a statement about what you think the evolutionary history of bacteria or a gene is. And a lineage is any part of that phylogenetic tree, so any branch of that tree with a bunch of organisms in it. And so the bacterial taxonomy as it stands is informed by the phylogeny, but doesn't always follow the phylogeny. So phylogeny actually happened in history. The taxonomy is what it is. It's an existing system, imperfect. Okay, and it took me, when I first came here and started working on microbiome stuff in 2009, it took me a little while to convince myself that making phylogenetic trees is difficult, but it's incredibly difficult. And the reason that it's so difficult is things can be, you can have 3 gene sequences. They can all be different by the same amount, but they can be different in different ways, and some of those ways are very informative phylogenetically, and some of those ways you don't care about. Like if one position in that gene is mutating very quickly, then that's not very informative for placing your sequence in the tree. But if one is pretty conserved and only changed exactly once in the evolutionary history of that organism, then that'll be really informative for the tree. There's no good algorithm to make the tree, and the number of trees is astronomical. So you can never get the best tree in general. You can get a pretty good tree by a number of methods. But there's like philosophical debates that people have about what's the best system for making a phylogenetic tree. For the purposes of this talk today, it doesn't matter so much. So we're going to be talking about phylogenetic trees that are made on one bacterial genus, where the amount of evolution is not like as much as if we were analyzing all bacteria who ever lived. So people will get roughly the same answer with fancy methods as they do with basic methods. And likewise later in a subsequent lecture, when we talk about diversity, in a lot of our bioinformatic pipelines, we're forming phylogenetic trees across all of bacteria to get unifrac distances between samples and to try to scale samples as being different from each other. As it turns out, imperfect trees, you can do kind of an okay job of producing a phylogenetic tree, and even an imperfect tree will be pretty informative. But the book I want to recommend is, if you're interested in this topic, the book on it is by Felsenstein, and you can read about the whole historical debate and all the fighting between different varieties of people who study phylogenetics. Okay, so this is an example of a phylogenetic tree. This tree was presented in a paper where they established 2 new species of Bacteroides. And so pretty much everybody living around Philadelphia has a fair amount of this bacteria in their guts. So we're all carrying around some of these species. And they've made this tree from the 16S rRNA gene. That's, I forget whether it's the most slowly evolving gene, but it's very slowly evolving. And every bacterium has it. It helps to form part of the ribosome. It's not translated. Okay, so here are the relevant pieces of information. Besides the tree, you see how the sequences from all these bacterial strains are phylogenetically related. This is their best guess. This is 1% sequence divergence. So on this scale, one base pair out of 100 is different between the 2 strains. They do some checking to see if they just sample some of the positions from the gene and then remake the tree, how stable the tree is with regard to the content of the gene. And so that's where you get these numbers. If you see low numbers on some of the nodes, then you wouldn't trust that branch very much. Okay, and then they want to give the tree a root, and there are several methods of doing that. And one method is to put in an extra strain of something that you're sure is outside of this genus. So you include one thing that's very different, and here they included a strain of Parabacteroides. And so they know wherever Parabacteroides is connected, that's the root. That messes up your tree sometimes, so there's a total art to doing this. And this tree contains 43 strains, and the number of possible phylogenetic trees is 10 to the 60s. So we're many, many, many orders of magnitude above Avogadro's number. Okay, and then in the same paper, they give a different tree for same strains, but different gene. And so I'm showing this just to emphasize that if you look at a different gene, it evolves at a different rate. You'll have a different guess as to the phylogenetic history of all the bacteria. Okay, so where do we get the names for bacteria? They're published in one journal. It's the International Journal of Systematic and Evolutionary Microbiology. And the critical thing about named bacterial species, they're defined with reference to a type strain. So there's one strain that people have mailed out to collections of bacteria, and that strain defines the species. If you're like that type strain, then you are in the species. If you're sufficiently different from the type strain, then you're a new species. And so there's new bacterial species that are published every month in this journal. They have to be genomically distinct from existing species. Their names are determined by making those phylogenetic trees and observing where they're placed. If they're placed within an existing genus phylogenetically, then they get that genus name. If they're outside, they get a new name.
I have a question. Is there like a hard cut or idea of what's sufficiently different to be a new species?
Yeah, totally. There totally is. It's on the next slide, I think.
Okay.
Maybe 2 slides up, but we'll cover it. They have to be, so one of the requirements, there's a genomic requirement that's the final arbiter. But you also have to be phenotypically different from existing bacterial species. So there has to be some set of lab tests that people can do on the isolate to distinguish it without sequencing its genome, which turns out to be a pretty low bar. It's easy for bacteria to be different. The website, so bacterio.net, has information on all the named species. My tip is search on the genus name that you're interested in, and the genus pages are very helpful. Yes?
So I know outside of bacteria you have cryptic species, which are genomically different but phenotypically the same, I believe. Does that exist for bacteria? Because there's like so much.
Yeah, good question. I can't cite an example of that. So, as far as I know, no, but yeah, theoretically it could happen.
How would you be able to say with certainty whether they were typically the same? Because it's like you can prove that they're different, but you can never prove that they're the same because there's like an infinite amount of tests. Because all bacteria don't require a single nutrient or anything like that.
I think in practice it's very easy to show that, or it's relatively easy to show that bacteria are different because you can have differences even within a species. And so that's usually not the deciding factor. It's more that you're trying to hit the marker for, this is the impression I get. I've never named a species. It's more that like you're trying to hit the genomic cutoff.
There is like a 3-page long list of guidelines that you have to establish with submission to this journal, though.
Yeah, and there's like a paper from 2010 that goes down all these guidelines for like when you know you've got a new species. Okay, so going back to our phylogenetic tree, these T's mean that this strain is a type strain for the species. And so here they've isolated 2 strains of this new Bacteroides species and 4 strains of this one. They select one of the strains, designate it the type strain, and they mail that out to at least 2 archives of bacterial strains. And when they do the phylogenetic tree, in these papers, they only include type strains of existing species because the goal is to place your new strain in the realm of existing species. So if they included other bacterial strains, maybe the tree would change and your answer would be different. You never guarantee that. Okay, here's the cutoff. So how is the 16S gene related to bacterial species? So one of the major approaches in metagenomic research is to sequence the 16S gene and use that as a marker for the bacterial community. So as it turns out, the final arbiter for new species in these papers, the guideline is 70% in a DNA-DNA reassociation experiment. So this is an old type of assay that looks at genomic similarity. And so if you're above 70 and everything else is working out, you're the same species. If you're below 70 and everything else is consistent, you're different species. The 16S gene can be the same between different species, but it's never different if you're the same species. So, except in very rare scenarios with like bacteria at hydrothermal vents and stuff like that. So it's sensitive, but it's not selective. So if you see a 16S gene and it's sufficiently different, if it's 3% different, you can be sure, well, I've got a different species here. But if you see similar 16S genes, it's up for grabs. Okay. Good. Oh, and sorry. And this is the paper that outlines the full set of guidelines for new species. And then how does this relate to actual genomic similarity? So we're in the age of genome sequencing now. How does the 16S similarity relate to the full genome similarity? So to address this, people have used a method called average nucleotide identity, because bacterial genomes can kind of get rearranged and some stuff can get dropped. And so with this scoring system for bacterial genomes, they don't pay attention to parts of the genomes that don't line up. So the recipe for this is they take two genomes, they blast them, they look at all the alignments that come out between the two genomes, all the local alignments, and they say whatever the average similarity is between parts that align. Okay, so this basically follows the same pattern. You see that that upside down L shape. So genomes can, the 16S gene can be similar, but genomes are a little bit different. But the 16S gene is rarely different when the genomes are similar. Okay, and just to round out this section, many bacteria are legitimately unnamed. So there is no good name for them. It's not the bacterium's fault. It's our fault for not naming it. This figure is from a paper. It came out in 2002 from the University of Pennsylvania, where they took mice and took all parts, small intestine, large intestine, fecal material, and they sequenced a bunch of Sanger clones. They got full-length 16S sequences. And there's this whole clade of mouse fecal bacteria that's totally unnamed. And they called it mouse intestinal bacteria. And it was that way until people started doing microbiome work, and Phil Hugenholtz put together another naming system called Green Genes, and he called it S247, this whole clade of bacteria, after one of the samples in this paper. And so that's been unnamed finally just last year in a big project. They finally isolated one strain from that clade. And so now we have one example that you can grow in the lab of this whole family of bacteria that can be about 80% of fecal bacteria in lab mice. All right, and other examples. So these Clostridium ASF 502. So ASF stands for altered Shadler flora, and that's a set of 8 bacteria that you can order mice with these 8 bacteria in them, and it's been used in research for 40 years in countless papers. Many, like 4 or 5 of the 8 species still unnamed. Segmented filamentous bacteria, so the groundbreaking work on small intestinal bacteria and the host immune system, where they're regulating Th17 cells. Still no name for those bacteria, or I think they named them, but then they named them wrong and they had to go back. But people don't use the name anyway because everyone knows them as SFB. And then if you look around here, Clostridium is the poster child for a poorly named bacterial genus. So it's all over the place on the phylogenetic tree and all mixed in with other genus names with seemingly no pattern phylogenetically of how the bacteria got their names. So these bacteria were named a long time ago before people could sequence 16S genes, and so they were named according to what people saw in their experiments. Okay. So we have all these issues with naming bacteria, and some bacteria don't even have names, and the names might be bad. So another approach might be, well, we have all the gene information and we can make these trees, let's just do it numerically, and then if we have bad names for bacteria, that doesn't affect our outcome. That's a great idea. The idea itself was introduced in 1963 by Sneath and Sokal, the same Sokal from your biometry textbook if you've taken biostats. And they weren't working with sequence data. They were working with like macroscopic organisms, as far as I know. For our purposes, and for most purposes these days, OTUs are sequence clusters. So we've collected a bunch of sequences. We want to put them into taxa. The term is it's very non-committal on purpose. So you don't want to say anything about the quality of the taxa that you've made. You're saying, well, I put these bacteria into groups. They're my operational groups. Maybe I did a bad job. Don't blame me. At this point, you might be wondering, well, we seem a little obsessed with putting bacteria into groups. Why are we so obsessed with that question? Why do we even have this goal? Let's just think about all bacteria as individuals, right? There are two reasons to put bacteria into groups. So to make comparisons between different environments or different samples, you'd like to have some common categories, some common foundation to compare bacteria. And that involves making groups. You can't get away from it. The other reason that, in particular, that OTUs are made with DNA sequencing is that it also kind of is supposed to get around this issue of sequencing error. So the DNA sequencers are producing errors in the sequences. Those produce some spread of from the true sequences. There's some variation just from the sequencer. And so the thought is, well, if we numerically cluster our sequences together, we'll put all of those error-prone sequences back together in one sequence cluster, and we'll get back to like the right answer. Everyone uses a 97% similarity threshold. It comes from that figure that I showed you earlier. So this figure is from the 1994 paper by Stackelbrandt. And the funny thing is, they say in that paper, the actual sentence that is written in the paper cites 97.5. So, but people rounded down. Maybe if I just take 97, then if I'm below 97, I'll be really safe. Okay. So clustering is another thing that that's somewhere between art and science. There are many ways to form clusters from the same set of objects, and you form the clusters with different goals in mind. And so here are the primary techniques in clustering that are used with 16S sequencing. The one that's probably most often used is, the one that's used in the Chime software is this one. So this is maybe the easiest to understand. So you line up all your sequences in a file, and you take the first sequence. You don't have any OTUs left yet, so you put that sequence in a new OTU. You go to the second sequence. Is it similar enough to the first OTU? Yes, I put it in there. Third sequence, is it similar to the first OTU? No, it's not similar enough, so I make a new OTU. And you just run through your file like that. It's called a greedy algorithm because it always takes that first sequence that it sees and makes that the defining sequence for each cluster. So these algorithms are really fast because you don't have to go back and resort your clusters, but the first sequence might not be in the middle of the cluster. It might be kind of on the edge. And so one outcome here is your clusters might be a little bit lopsided. Okay, and you might not pick the good centers. Other methods of clustering, so these methods are agglomerative. They're used more in if you use the Mother software by Pat Schloss. So he had this kind of had this idea that you should do a full alignment of all your 16S sequences from the experiment, get a similarity between each, build a big distance matrix between each pair of sequences, and then do agglomerative clustering. So when you start out, they're all in different clusters, and then you put them together in clusters according to one of these three methods. So you can require that the average distance between points in both clusters is less than 3%, for example. You can require that all the points in each cluster are similar to each other. That's called complete linkage. Or you can say, well, only one pair of sequences needs to be that similar, and I'll let the cluster spread out as long as it connects to at least one sequence in the cluster. And so that's called single linkage. Here you tend to get long stringy clusters, but you're never splitting up a good cluster that's coming in under your threshold. Here you might split up legitimate clusters, but you're erring towards making tight circular clusters that are very consistent.
So Kyle, I think you're going over kind of the advantages and disadvantages of each method. So if we're interpreting data or looking at it, is there like a industry standard for what is considered the best or most appropriate for each situation or scenario?
Yeah. I think the way it plays out, so I typically use this one. Because we'll see this in the demo, but you should be clustering at a similarity level that's not critically important to your study. So if you use one method or another, you shouldn't get a totally different answer. And if you're worried about that, you should investigate that. No way around that. I forget which is recommended in Mother. I do know that a lot of approaches start with this, and this was an idea that came out of Mitch Sogan's lab and is in some other pieces of software. So this will it'll never split up a good cluster, so your clusters will be too big. So you do this first, and then you go within each cluster and you use some fancier algorithm to try to say, should I split this up?
So how do you choose a representative routine for each cluster?
Yeah, great question. So these methods.
That arbitrarily defines the first one that it comes to, right?
Yeah.
And is there any checking of where that kind of is in the cluster afterwards, or it's just done?
No, it's done. And you generally want, there's some justification for keeping that sequence as the representative, because even if it's not in the center, it was the selected sequence for that cluster. Here you can do things like use the most common sequence for the cluster, but that might be on the edge. Right. Typically it's not, because if it is the most common, you have errors that are kind of circular around that sequence. There are also methods to find the center sequence in the cluster, though I'm not sure how widely that's used.
Okay.
I'll talk about a couple pieces of software and how they actually work that might answer that better. Okay, so if you start to look at the differences between these methods, you get into some really weird and tough territory. So these alluvial diagrams are about as good as it gets for really explaining this and graphing it. It's tough. You don't know what the right answer is, so you don't know how the bacteria should be clustered. So you're comparing two clusters, and you're looking at, well, what clusters were split, what sequences were in common, what percentage of this did I have, how many clusters did I form? That's different too. So here's an example of one of these analyses, and it's not easy to interpret, but generally, if you look at the complete linkage, it's at the same similarity level. It's making more clusters because it has a more stringent requirement for each cluster. And if you look at single linkage, well, anything that's connected to at least one sequence in there can be in the cluster, so it tends to form fewer clusters. That's not always the case. It depends on the distribution of your sequences. Another thing, just to kind of point out from this, is that these are two pieces of software using the same general, very general types of algorithm, and they don't form identical clusters, and the number of clusters is much different between those two pieces of software. So you hope that at the end of the day, this doesn't adversely affect your results critically. All right, so here's the software. As far as I know, Doder kind of started the this kind of area of software with like the 16S business. So that's Pat Schloss's software. That's a predecessor to Mother, which you can use today. It's a bioinformatics pipeline. But again, Pat's idea was, well, we'll make a giant distance matrix from all the sequences, and then we'll use agglomerative clustering. And this was a real computational challenge when this software was written, just to hold the distance matrix in memory. All right, CD hit is like one of the standard pieces of software. So this came out a year later. This is where they introduced the greedy clustering algorithm. We just take the first thing that isn't similar to any existing OTUs, and we make that the representative for a new OTU. I do want to point out that this method is pretty much the same method by which bacterial species are named. So, right, if you have a new strain in your lab, if it's not similar to an existing species, you name it the new species, and that becomes the type strain. And it's order dependent based on how people did their research.
Actually, Kyle, can I ask you another question about CD hit and uclust? So,
yeah.
read 1 becomes OTU 1.
It becomes the representative for OTU 1.
So now, read 2 is, let's say it's greater than 97% similar with read 1. It gets put in that OTU.
Yep.
Now we're going to read 3.
Yeah.
And it's only compared against read 1.
Yep.
Now, what is the probability that it's, say, less than 97% similar to read 1, but is more than 97% similar to read 2, which it's not compared against?
Yeah, I don't know. I mean, it totally depends on the distribution of those reads. And yeah, you get into situations like this, like they're illustrating in one of the papers. Like this should probably be your cluster right here, but you've done a good job choosing the center, but it's just that your radius is slightly wrong. And the correct radius is probably different, slightly different for each cluster. And so these sequences end up being centers of new clusters, even though if you were looking at it, you would probably draw a circle around the whole thing. Okay, and so people have worked on this problem up until now. So VAMPS was the idea where they would do single linkage clustering first. They'd make clusters that were too big, and then they would go and look at those clusters to see, should we split these up? And as far as I remember, that came out of Mitch Sogan's lab. Then uclust was a very fast implementation of this greedy algorithm. Chime started using that, and they wanted to use something that was open source, so they wrote SortMe RNA. And then people said, well, why don't we just use fancier methods to try to get better clusters? So what if we have to wait a little while? So some people wrote some software to use Gaussian mixture models and really try to optimize this. MPIC was a piece of software that tries to address this issue by varying the cluster size to try to optimize that. Oligotypes I'll have a slide about. But swarm is this figure is taken from the swarm paper, and that's another attempt at kind of single linkage clustering and then splitting them up, except they made it really fast. So Swarm is one of the good pieces of software, and in parallel, there's kind of been another effort to say, well, why are we trying to do species level clusters? That's a hard problem. Let's just try to fix the sequencing error and not concern ourselves with the biology of bacteria. So we'll just tackle the technical problem. And that was an effort that started around 2009 with Chris Quince's amplicon noise software, Pyronoise it was called back then. But the issue with those pieces of software is they took a long time to run. That was okay. You could deal with that if they got good answers, but they didn't agree with each other. This is a plot from the Acacia paper and shows that this is dealing with substitution errors, and so they're trying to figure out if this was a sequencing substitution or if it's a legitimate substitution in the 16S sequence. And they only agree on 178 out of potentially 20,000 substitutions that they analyzed. So this is comparing 3 pieces of software. And another thing that kind of happened once this stuff all played out is that you might do the analysis with one data set and then combine it with another data set and do a combined analysis and remove errors, and you'd get a totally different profile, which is natural because you're trying to estimate the errors from the data and you've given it more data so it has more information. But it kind of destabilized the results from those papers. So I stopped doing this around 2012. But it's back. Denoising is back. In the last year, 3 new pieces of software were published in this area, and some of them are tuned to Illumina-specific error profiles. Some of them are just trying to generally solve the problem. It's by the nature of the problem, it's hard to separate the sequencing error from the natural variation. And you're always trying to do it. You're trying to make your estimate from the data, so your estimate's always going to change if you change the data. So if you add data, your estimates are going to be different. All right. There's another attempt to go below the OTU level that I thought was really interesting. Murat Aaron published this in PNAS in 2014. So their idea was we'll take sequences that are pretty similar to start with. So we choose a genus first, like Streptococcus. Then we take all the Streptococcus sequences and we put them in a multiple alignment. And we can align them pretty well because they're all pretty similar to each other. So there aren't any of these weird alignment artifacts we don't have any tough decisions. And then we look down the columns of the multiple alignment and we try to score stuff. So if all the nucleotides are the same down that column, then it gets a score of 0. Likewise, if they're all the same, but just maybe one sequence is flipped to T, it gets a really low score. But if it's like half A and half T, then it gets a really high score. And we want to pick out columns like that. They're informative for differentiating species. So there's one step in this process which is manual. So you have to look at this plot and select what your level of information is going to be. And once you do that, you have a small number of columns here too from the multiple alignment. And this one can either be T or A. This one is G or C. And so then you have either AC or TG, and those are your like subspecies or strain level oligotypes, as they call them. So this is they're trying to get down as far as they can go with the 16S gene to differentiate bacterial groups. And as far as I know, this seems to me the most successful way of doing that. All right, and finally, we're going to touch on the issue of reference-based operational taxonomic units versus de novo or from scratch OTUs. So you might have the idea that because there are millions of 16S sequences already deposited in databases, maybe we should just check our sequences against the sequences that are in the database, and we could probably form really good clusters from those full-length sequences that are in the database. So we should use those and then put our experimental sequences into existing clusters. The two problems. So the advantage of that is that it's stable. So if you have the same database, you'll always have the same clusters. So you have the same set of buckets that you're putting your experimental sequences into, and that way you can compare experiments really easily because you've used a common set of categories for all your experiments that you're analyzing. So that's why you would want to do that. Oh, and maybe you can use the phylogenetic tree from the full-length 16S sequences, and that'll be a really good phylogenetic tree. So here's why you might not want to do that. So disadvantages. The first disadvantage is that not all of your sequences will be represented in the database. So you're only able to place a certain percentage of your experimental sequences into the existing buckets. All right, but what if your percentage is good? What if you're placing like 98%? Then you run into issues that you're sequencing a small section of the gene. Your database is made from the whole gene, and so you might be able to legitimately put your segment into any of 3 or 4 buckets. So how do you pick which bucket to put it in? So you're back to this old clustering problem. There's no good answer. You don't have enough information to decide. And you wanted to use the really good phylogenetic tree, but you don't know where to put your sequence in the tree. So you can do the reference-based methods and the websites, like if you submit your sequences to the Cheetah website for analysis online, they'll use the reference-based methods. But just be aware of this issue. You could place into any of several existing OTUs, and you can't always tell ahead of time which one you're going into, and they might be named differently. Okay. So we're going to finish off with the process of naming the sequences. So we've covered how real bacteria names are generated, how to numerically cluster sequences into taxa, but they don't have any names yet. So how do you put names on them? So you're basically comparing your taxonomy that you made numerically to an existing taxonomy that someone else came up with names on it, and you're trying to place your taxa in their taxa. Okay, so the two popular approaches. So the RDP classifier was the first piece of software to really tackle this in a great way, to my knowledge. And the way this works is it selects a short sequence length. I think it uses, I forget whether it uses 8 or 11 base pairs. But it uses some really short sequence. And then it finds in your experimental sequence or in the reference sequences, it finds all those short subsequences, like you see over there on the right. And it builds up a big lookup table based on those short sequences. So it says, for every genus, I know how many times this short sequence appears in that genus. Those are called K-mers because they're like, these would be like 8 mers. Or if you used a sequence of length 7, it would be a 7 mer. So K is like the stand-in for the length of word. Okay, so you've built this giant table. Now you go through your sequence and you look up each word in the table, and that gives you a probability for that sequence being in that genus based on the amount of matches that you get. And so you solve a probability equation, and then you do some like resampling from the reference, from your query sequence to come up with a confidence on that. So there's no alignment, and computers are very fast at doing these lookups. Because for each word, you can have a number, and then you know right where to look. You just say, I have a big array of this stuff, and I'm going to look at position 101. Computer, go do that. That's really fast. Okay, so the other way is to actually build a search database from all your reference sequences and align the query sequences, find the best matches in the database. So you can use BLAST, or there's many other pieces of software that kind of solve this problem. 16S sequences are very similar to each other, relatively speaking. So in the world of this type of software, performance differences are not super critical between different pieces of software. It's relatively easy to do the search. So you search a database, you find some number of top hits, and then you go through some kind of voting method to say how much do they agree with each other. So the default method in QIIME is look up the 3 top hits using the uClust software, and then if you have 2 out of 3 votes, then you name it that thing. So you could have one bacterial hit that has a completely different name. It doesn't matter, you'll name whatever 2 out of the 3 vote for. The old method is to just search with BLAST and take the top hit. So you can totally set n equals to 1 here. All right, and so here are the taxonomic systems that people use when working with this data. So the RDP taxonomy is the oldest one. Everything has a genus. The dead giveaway for the RDP taxonomy is that you'll see these Clostridium cluster numbers. The green genes taxonomy comes in QIIME by default. So this is a system, to use the RDP classifier, it expects everything to have a genus, but a lot of things in green genes didn't have genus labels. So the solution that we came up with was we'll put blank labels in there. So we'd totally not commit to any name. But then as a result, you can kind of hit conclusively to a blank label, or you can not hit very well to any labels, and there's kind of a weird difference between that. And I usually don't pay attention to that, but just be aware that pops up in papers. You'll see sometimes that something's named blank. It's because we had to put in blank labels in this taxonomy to make it compatible with this. Also, you'll see taxa in brackets coming from here, and that indicates that it's a part of the genus that is different than the main part. So there's some species of Prevotella that are phylogenetically different from the main cluster of Prevotella, and they're named Prevotella in brackets as punishment for being different. And the Silva taxonomy is another version. So I think Mother uses Silva by default. And so this is a really old project. Comes out of it's on the European side. Comes out of the ARB analysis software, and they hold a browser for all the taxonomies, so you can see them compared to each other. And then there's the NCBI taxonomy, which is mostly generated to hold sequences in GenBank. And so that's its main thrust. It has buckets for everything you would need a bucket for when you're depositing sequences in their system. So it's not meant to they keep it pretty well maintained, but it's not fashioned exclusively to classify organisms. It's also it has human gut metagenome if you need to label something that. All right, and Pat Schloss had this great quote in one of his papers from 2010. So when we do 16S sequencing, we're sequencing a short segment of the gene, and so we're sequencing a marker of a gene, which is itself a marker of the genomic diversity of the community. So we're kind of twice removed from what we really want to be studying, and 3 times removed because we really want to be studying what the bacteria are doing, what they're capable of doing. And so we're observing the genome as a stand-in for that, and then we're observing a piece of the genome as a stand-in for the genome, and then we're observing a piece of that gene in our actual sequencing. So these are the people who are involved in the PennCHOP microbiome program, many of them here, so please say hi.